Okay, this video lecture will introduce you to the simple Mendelian genetic principles and how to use a Punnett square to predict the outcomes of different kinds of crosses. We'll satisfy the requirements for learning target number 1, which states I can apply the terms phenotype, genotype, allele, homozygous, and heterozygous in the context of a monohybrid cross. To begin talking about genetics, we need to really start with the father of modern genetics. It was a man named Gregor Mendel. And Gregor Mendel worked with a particular subject, research subject. He worked with peas. And the reason that peas were particularly useful is because they had several traits that were easily identified, and they seemed to come in only 2 varieties. So plants were either, tall or short. They were purple or white. The peas were either round or wrinkled, or the pea pods were green or yellow. Anyway, they had these very definite traits, and as Mendel bred them, caused them to reproduce, and then grew their offspring, he observed some tendencies in the way that they passed on their traits. And when he explained them, and he used mathematical terms, he used probability, when he explained them, he uncovered the way that organisms pass on their traits, and I'm going to show you a little bit about how he did that right now. Gregor Mendel's experiment essentially occurred in 3 steps. He began with 2 purebred parents for opposite traits. So in this example, we are looking at a pea plant, and the trait that we're looking at is whether they are tall or short. And he took plants that were purebred, they always bred as tall plants, and he took plants that were purebred short, they always bred as short plants, and he crossed them, he forced them to reproduce. And then he took the seeds, the offspring of that reproduction, and he grew them, and what he noticed is that both plants appeared tall. The dwarf plant, the dwarf trait, had essentially disappeared. Now, at the time, he didn't use the letters capital T, or lowercase t, lowercase t. He simply observed this. It was later on that we assigned those words. What he said is, he said the allele for being tall appeared to be dominant. So then he took those 2 plants and he crossed them, meaning he reproduced them. And when he did that, when he grew their seeds, he came up with a different ratio. Out of every 4 plants, 3 of them, or 75%, remained tall, and in one plant, the dwarf trait, or the short trait, reappeared. So this is in the 2nd generation of offspring. And this sort of puzzled Mendel, because it appeared that this trait of being short had disappeared in the parents here in the 1st generation, yet reappeared in the 2nd generation. So he needed to come up with an explanation for this. And genetics gives us a tool for understanding this. So we're going to look a little bit more closely at how Mendel did those crosses, and we're going to be using Punnett squares to understand them. So, looking at his first cross, and when I say cross, I simply mean reproduction. So in his first cross, he took purebred parents. One was purebred with the phenotype tall, the other was purebred with the phenotype short, meaning their genotype, which we know now, Mendel didn't know this, but what we know now is that they're capital T, 2 dominant alleles, and the short parents were lowercase t, lowercase 2, they had 2 recessive alleles. We use the word homozygous to describe them. And there's the definition of that word, keyword down there. So let's look at what Mendel did, or what essentially happened to explain why all of the offspring came out looking tall. So this is a Punnett square right here, and this is a tool you can use to predict what parents would have looked like based upon different kinds of offspring, or what offspring will look like with different kinds of parents. And anytime I ask you to set up a Punnett square, you should make a gridded box that looks something like this, and you should know that the parents are always going to go on the outside, and the offspring are going to be represented in the middle, and it'll give us a way to predict the likelihood of certain kind of offspring. So if we go back again, and in that example I just gave you, I said Mendel took a purebred tall plant and crossed it with a purebred short plant. And what he got, so capital T, is parents that were, parents were tall, parents that were short. And so what was happening was the parents with the two tall alleles were dividing up their two copies, their two capital T's, because every organism has two copies of each gene, two alleles. And the short parent was doing the same with its alleles during the process of meiosis to create sex cells that might be lowercase t or sex cells that might be uppercase T. When the reproduction occurred, this, which could be the sperm cell, and this, which could be the egg cell, would join together to create capital T, lowercase t. Or these two could have joined together to create capital T, lowercase t. What I'm showing you here is what is called random fertilization. We don't know if this one or this one is going to create the offspring, but it will be one of those two. So these two would create the offspring, and so on, and these two here. So the end result is that all of the offspring were capital T, lowercase t, meaning they were all tall. So in this case, 100% of the offspring were tall. All of the offspring had one capital T allele and one lowercase t allele. That means they were all heterozygous. They had one version of each trait, or one version of each gene, excuse me. And so there we have an explanation for why all the offspring were tall. Because they were all heterozygous, they all had one dominant allele, a capital T. And because dominant alleles suppress the appearance of recessive alleles, all of the offspring looked like the dominant trait. They looked tall. So the next thing that Mendel did was he crossed those two individuals. And to kind of show you how that's set up, he crossed the second generation. He crossed two tall parents, phenotypes of being tall. He took, so their genotypes were capital T, lowercase t, one of each allele. These individuals were heterozygous, and there's the definition of that, and I'll show you what that looks like on a Punnett square. So here we have our two heterozygous parents. Remember, they have two copies of each gene, each one does, and during meiosis those are going to separate into sex cells, so we will have one parent that would be able to contribute a capital T or a lowercase t. Okay, that's this parent, and then we'll have a parent up here that would be able to contribute a capital T or a lowercase t, and the process is the same as I showed you before. Those two sex cells could have joined together, those two sex cells could have joined together, and so on, which means the offspring possibilities are capital T, which would make a tall offspring, capital T, lowercase t, heterozygous again, capital T, lowercase t, or lowercase t from these two up here. Okay. So in that cross, the short or dwarf trait reappears. We have three individuals that show the dominant trait. Two of them are heterozygous, carrying one dominant allele and one recessive allele. One is homozygous dominant, carrying two dominant alleles, and one is homozygous recessive, carrying one recessive allele. And by this process, Mendel was able to explain, using percentages, what happens to each of the genes. He figured out that the genes had to separate. Okay, this was before we knew that sex cells were created, but he knew that the genes had to separate, and then he knew that the genes were recombining during sexual reproduction. Without knowing anything about meiosis or really about how sexual reproduction actually recombined genes, he was able to describe the process, and he was so accurate that we still use his explanation today, and we use the Punnett square as a way of demonstrating that. Now, as you've been following along, if you've been up to this point copying down the Punnett squares as I've been doing them as demonstrations in your notebook, that's great. If you haven't been, please go back and do so that we can talk about some of the kinds of questions I might ask you. So let's look at that second cross that we did with two heterozygous parents. These are questions I might reasonably ask you about that cross. What % of the offspring are heterozygous? Well, out of 4 individuals, 2 of them were heterozygous, which means 50%, 2 out of 4 were heterozygous. What % are homozygous dominant? Look at your sheet, answer the question. Hopefully you answered 25%. The one in the upper left-hand corner is capital T, homozygous dominant, so 25%. What % are homozygous recessive? Again, I hope you answered 25%. The lower right-hand corner, you'll see lowercase t. That individual is homozygous recessive for the trait. What percent have the dominant phenotype? So this answer is a little bit different because you're not describing what the genotype is, you're describing the phenotype. What do they physically appear like? And in this case, phenotype, the dominant phenotype is to be tall, and 3 of them are tall. The upper left, the upper right, and the lower left are all tall plants with the homozygous dominant and the 2 heterozygous individuals. So 75%, or 3 out of 4, will have the dominant phenotype, and that leaves 1 with the recessive phenotype, 1 out of 4, which is 25%. These are the kinds of questions that I would reasonably expect you to be able to answer about simple Punnett Square examples. And next I'm going to show you a few more complicated examples. What I think you should do is I think you should look at the question and then pause the video, try to answer them on your own, and then listen to my explanations of each. So here's the first. If we have purple flowers, capital P, dominant to white flowers, lowercase p. This is just a true statement. Purple is dominant to white. If a homozygous dominant purple plant is crossed with a heterozygous plant, what percent of their offspring will have each phenotype? Why don't you pause the video right now and try that practice problem, and then I will show you how to answer that problem using the Punnett Square. Welcome back. When you set up your Punnett Square, you should have started with a homozygous dominant parent and a heterozygous parent. Okay, both of them will appear purple. Okay, each of these alleles will get separated during meiosis, so we will have the homozygous dominant parent will separate into a capital P and a capital P, and the heterozygous parent will separate into a capital P and a lowercase p. Okay, and after we do the crosses, we are going to have capital P lowercase p here. Okay, capital P lowercase p here, capital P here, and capital P here. And the question asks, what percent of the offspring will show each phenotype? Well, the 2 possible phenotype options are white and purple. Now, you can't see the color on here very well, but hopefully you can figure out that since purple is the dominant trait, having 1 allele for purple will cause the offspring to be purple. In this case, this will be purple, so will this, so will this, so will this. So all 4, that means 100% of the offspring will be purple if the 2 parents had these traits. Okay. And now, I mean, we're using this with pea plants, but you could apply this to any trait that has a dominant recessive characteristic, and sometimes people use this to predict the outcomes of their own crosses, which is what we'll focus on more in class. I'm using Mendel's experiment here to illustrate it, and in class we'll use other examples. Let me show you one more. Try this practice problem again with purple flowers and white flowers. In this case, I'm giving you the outcome of I'm telling you what the offspring look like and asking you to figure out what the parents look like. And this actually models more closely what we're often interested in, which is what are our parents' genes based upon what the children's genes are. So try this problem and I will explain it in just a moment. Okay. So here's how it sets up, and this is a bit of a logic problem, but we know that 2 of the offspring were white, so they had to be lowercase p, which means they had to get it from each of their parents. So one of their parents had to have been white in order to create 50% of the offspring that were white. However, in order to have purple offspring, the other offspring would have had to have been heterozygous for the trait, because they would have gotten a lowercase p from this parent over here, but they weren't themselves white, so that means they had to get a capital P from their other parent, making 1 parent heterozygous for the trait and 1 parent homozygous recessive. If you didn't understand that, go back through that again until you understand it. It's a logic problem.